Haddock. I'm a sophomore chemistry major working with Dr. Guy Caldwell in the Department of Biological Sciences. Today, I'll be presenting on the impact of RNA editing on pcca-1 function in an Alzheimer's model. Alzheimer's disease is a form of dementia characterized by the pathogenic protein amyloid beta, which is chiefly expressed in a specific type of neuron called glutamatergic neurons. This dementia affects an extremely diverse population of people with no clear evidence yet established that explains the onset of the disease. While genetic factors are a risk in developing Alzheimer's, there have been cases with identical twins in which one twin develops Alzheimer's while the other twin doesn't. Because of phenomena such as this, a rapidly expanding field of Alzheimer's research is epigenetics, or the role of environment on gene expression. There are many cellular mechanisms that control gene expression, which are reactive to environmental conditions. One such mechanism is RNA editing. Before I explain RNA editing, I will quickly review how proteins are made in the cell. So DNA of the genome is transcribed into RNA, which is translated into proteins by ribosomes. And DNA and RNA are both made up of single-letter nucleotides, the order of which determines which protein will be made. Sometimes after DNA is transcribed, the RNA undergoes single nucleotide edits before being translated into the protein. In humans, this is done by the ADAR enzymes. And what they do is they convert A nucleotides into I nucleotides. Then, when that edited RNA is read by the ribosome, the I is interpreted as a G. So it's functionally an A to G edit in the RNA that could result in an altered protein product than what is encoded for in the genome. The level of activity of these ADAR enzymes can vary, and studies show that there are lower levels of RNA editing in Alzheimer's brain samples than in healthy brain samples. This could mean that there is a higher incidence of incorrect proteins being made in Alzheimer's brains because of the lack of editing. One gene whose RNA undergoes editing in healthy brains but not in Alzheimer's brains is pcca. The protein product is shown here, and it functions in the mitochondria to create important intermediates for several metabolic pathways. Here in the red is the edited protein made in healthy brains, and in the blue is the unedited protein made in Alzheimer's brains. And there is a significant structural difference between the two, and this could mean that the Alzheimer's protein is not functioning correctly. While pcca has been established as an important protein for overall mitochondrial health, there is no specific evidence on its role in neurodegeneration. So I aim to explore the neuroprotective effects of pcca and determine if these effects are dependent on RNA editing. To do so, I'll be using a C. elegans Alzheimer's model. C. elegans are a microscopic roundworm with similar cellular level organization to humans. They have neurons, which makes it possible to study Alzheimer's, and they have many homologous or synonymous genes to humans, one of which is pcca-1, which is the worm version of the human pcca. C. elegans also have their own RNA editing mechanism, which is catalyzed by ADR1 and ADR2 instead of the human ADAR, but the mechanism works the same way. The Caldwell lab has genetically altered C. elegans to express amyloid beta in glutamatergic neurons. And this is paired with expression of green fluorescent protein so that the neurons glow and we can analyze their structure. To recreate the human RNA edits to pcca in the C. elegans, CRISPR-edited animals have been created that have single A to G nucleotide edits in the pcca-1 DNA. These edits correspond to the A to G edits made in the pcca RNA in healthy human brains. This way, every time the pcca-1 DNA is transcribed, the edited RNA is made, and because the edits are hard-coded into the DNA, the presumably healthy protein is always made. This allows me to study the effects of consistently producing the edited protein on neurodegeneration. These CRISPR edits were made in a wild-type C. elegans, so I need to incorporate that into the Alzheimer's model. To do so, I will follow the following mating scheme. So CW12 is the CRISPR-edited strain, and I first need to mate that into DA1240, which is a strain expressing GFP in glutamatergic neurons. This gives me a non-Alzheimer's control. I will then mate that to UA198, which expresses amyloid beta in the glutamatergic neurons. This gives me the Alzheimer's model. I mentioned earlier that C. elegans have their own RNA editing mechanism, and while the mechanism is conserved across species, the editing sites themselves are not. So I don't predict that the native RNA editing in C. elegans will interfere with the study of this gene. However, just in case it might, I have ADR1 and ADR2 knockout worms that don't have RNA editing enzymes. And I will mate that into my two resulting strains to get an RNA editing free version so that I can cross-check and make sure that there has been no interference. In addition to looking at the RNA editing aspect of this gene, I also aim to quantify its overall role in neurodegeneration. To do so, I'll be looking at two extremes. On one side, I have the strain RB1774, which has a pcca-1 deletion, and it makes none of the protein. Here, I have a pcca-1 overexpressing strain that has extra copies of the gene, and it makes more of the protein than what is standard. I will mate both of these strains into the Alzheimer's model using the same mating scheme I outlined earlier, first into the GFP strain and then mating that into the amyloid beta strain. Once I have the strains made and ready for analysis, I will score their glutamatergic neurons and compare the results. The purpose of the aforementioned GFP expression is illustrated here. So in this amyloid beta and this GFP and amyloid beta model example, on day 3 there are 4 glutamatergic neurons, and on day 7 there are 3, so 1 has degenerated. Sometimes it is that simple that a neuron just disappears and that counts as degeneration, but I will also be looking at a whole range in between and considering factors such as each neuron's size, shape, structure, dimness of the GFP, location, connectedness to other neurons, and all of these aspects will be graded on a strict rubric to ensure that I score each worm consistently. Another way that I will analyze the glutamatergic degeneration is by testing the worm's mechanosensory response, which is done with a touch assay. So a worm is simply brushed with a small bristle, such as an eyelash, and if it moves away from the stimulus, that means it has functioning glutamatergic neurons and that its reflex is working correctly. However, if this worm had continued in the same direction, that means that it didn't feel the stimulus because its glutamatergic neurons aren't functioning correctly. For this assay and the previous assay, trials will be performed in triplicate with 30 worms scored per experimental condition. So this concludes my overview of this project so far. This semester, I have created the integrated transgenic line that overexpresses pcca-1. I have received the CRISPR-edited strain, and I'm beginning to mate that into the Alzheimer's model, and I'm about halfway through the other matings I showed. So throughout next semester, I will finish creating the strains and then begin data collection and analysis. Thank you. I will take questions now.
How long did it take you to create the transgenic line?
To create the...
How long did it take you to create that?
The strains?
Yeah.
So each mating takes about 2.5 weeks if everything goes correctly. And if something doesn't go correctly, then you're kind of set back another 2.5 weeks. So because these are done in succession, there's no way to get to this point without first coming through here. So this whole process can take 2 to 3 months. Yes.
So most of the assays that you mentioned today dealt with the structure of the neurons. Is that kind of the definition of neurodegeneration, or could the, let's say, quote unquote, degeneration occur due to something that doesn't quite affect the neural structure?
Yeah, so I am kind of looking at both aspects here. So the actual structure of the neurons is visible through the GFP expression, and if there's something wrong with how the neuron is, with its structure, then that does mean there is degeneration. The good thing about this touch response assay is that it doesn't really have much to do with the structure of the neurons themselves and more with their function. So if there was some kind of degeneration that we weren't able to spot when looking at the GFP expression, then it would come through in how the worm's reflexes are functioning.
So could you go into a little more detail about why genes undergo RNA editing?
Yes. Okay, so the true evolutionary purpose of RNA editing is still being explored, but one commonly accepted explanation is that this is evolved as a defense against an autoimmune response. There are certain RNAs that, when transcribed, have a sequence that is complementary to itself, and they fold up on themselves to form a double-stranded RNA. And this resembles viral RNA and triggers an immune response. So if those RNAs undergo single edits such that they can no longer bind to themselves, it will prevent the autoimmune response and decrease the stress in the cell. And then over millions of years of evolution, those RNA edits just became integral to the structure of the protein. In a more immediate sense, what actually triggers an RNA editing event in a cell is kind of an open question. There are certain environmental factors that have been looked into, such as extreme temperatures causing higher levels of RNA editing, but that is definitely still an area that needs more exploration.